So chapter 5 really talks about, we're going to start getting into cellular functions now. We talked about parts of the cell, and we defined on what they are and basic function of what they do, okay? But now we're going to talk about functions that of parts of the cells or what the cell does in its normal life, okay? But first, the first function we're going to talk about is transport, moving things in and out of the cell. All right, but before we talk about transport, let's review, and I think this is a review because we covered the structure of the cell membrane generally when we talked about phospholipids. Yes? That sound familiar? Okay, not making stories up, all right? But let's general review of the structure of cell membranes. They are made of phospholipids, correct? And phospholipids we drew like this. Yes? And what are the lines? Tails that are composed of what? Fatty acids. Are those fatty acids polar or nonpolar? Nonpolar fatty acids, okay? So they are right here, nonpolar fatty acids, right? Fatty acid chains. So then what's this up here? That's what, Abby?
The phosphate head that's polar.
So this is a polar phosphate head. Right, and we could always put the little plus on there if we wanted to. Okay? So that's the basic structure of the cell membrane. And this part, this nonpolar part, is what? Hydrophobic. And this polar part is hydrophilic. Okay? That's the majority of the basic structure. That's the foundation structure of the cell membrane. But that's not all that's there, right? What else is there? So we're going to minimize that and get it out of the way. We have our phospholipids, right? Our tails. I'm just going to draw them like this. And assume that the black line is continuation of the phospholipids. We're zooming out a little bit. What else is in this cell membrane? Proteins. Okay, so we have proteins. We have some proteins that span the whole membrane. Right? We have some that are on the inside, and we have some that are on the outside. These proteins that span the whole membrane, they are called transmembrane. Really, they are integral proteins, meaning that they are integrated across both regions, right? Both the hydrophilic and the hydrophobic regions. They span the membrane. These other proteins that are on the either on the inside or the outside, these are peripheral proteins. Peripheral. All right. And there are other parts to this. Okay, we also have a lot, especially on the outside, the cell membrane has what's called the glycocalyx. Right? These are chains of carbohydrates. What's this? This is what's going to be called the glycocalyx. We'll talk about it in a little bit, but a glycocalyx. Okay? That's going to be composed of both glycolipids and glycoproteins. Right? Glyco refers to sugar, right? So that's a sugar or a carbohydrate. Glycolipid is a carbohydrate that's linked to a lipid. And we also have glycoproteins, which is a carbohydrate linked to a protein, all right? So we have these proteins, and collectively, this is what we end up calling what? What's the model of our cell membrane called? No. It's a phospholipid bilayer, but it's not just one part. It's not just one thing, right? A fluid mosaic model. What's a mosaic? Right? It's made up of different parts. And fluid, it is a picture made up of different parts, right? Or a piece of art. Fluid refers to the fact that it is dynamic. It changes. May.
What does that say about glycogen?
It's change of carbohydrates.
So all of that is a fluid mosaic?
All of this whole thing, this whole thing represents the fluid mosaic model. It's fluid because it can change. Mosaic because it's made up of proteins and phospholipids and some carbohydrates that are linked together to do a certain function. Fluid mosaic model. Okay? And the biggest function of a plasma membrane is to provide the boundary outside versus inside, and let things in and out based upon the structure. Okay? We know that this area in the middle, between the two bilayers, is hydrophobic. So it doesn't like polar things. It doesn't like charged molecules. It doesn't like big molecules. This leads to what we call the selective permeability of the membrane. Selective permeability states that some things get through and some things don't. Okay? Some things that don't get through are things that are too big, things that are charged, things that are polar, because of that middle hydrophobic layer of the phospholipid bilayer. This is what it looks like. It looks like a side of a mountain in western Massachusetts right about now, right? So what do you see there? Lots of color, right? So here's this right here is your phosphate heads. These are your phosphate heads. Okay? And then here, these green things are your fatty acid tails. Okay? What you have out here then, this right here is a protein. Do you see the coil? You kind of see the coils? All right? That's a protein. And then everything out here is what? H2O. All right? That's a pretty good drawing. So, here is your hydrophobic. I'll just put PHO. Okay? And then out here is your PHI, hydrophilic. Okay? Hydrophobic versus hydrophilic. All right? And you see all those little red dots up there are actually water molecules. So that's a space-filling model of what it looks like, of the phospholipid bilayer looks like. All right? But we know that phospholipids have two ends, right? A polar end and a non-polar end. And for that, they're actually called amphipathic molecules. Amphipathic means both two sides. Okay? So when you put phospholipids by themselves in a water solution, they self-assemble into this bilayer. All right? And so it exists as a boundary between two aqueous compartments. The inside of the cell being an aqueous compartment mostly made of water, and the outside of the cell being an aqueous compartment mostly made of water. All right, so this is a really good diagram of what the fluid mosaic model looks like. Okay, here are the two layer. Let's go with this one. All right, here's the two layers. Here's one phospholipid bilayer. That's the extracellular side, and then this is the interior side. All right, so we actually can talk about the bilayer having different faces, having a cellular face or being on the inside portion, and an extracellular face being on the outside portion. All right. And then in between those, you have your proteins that are integral or peripheral, and then you have carbohydrates that collectively make the glycocalyx. All right. One thing I haven't mentioned yet is cholesterol. You see, cholesterol is this thing right here. No. All right, is right here. Okay? And there, it's kind of embedded in here at different points. Okay? Cholesterol's function in membranes is to prevent these fatty acids from being too tightly packed. All right, so it kind of embeds in and prevents it from being too tightly packed. If it's too tightly packed, what ends up happening is that these membranes become too rigid and they can break. So cholesterol helps maintain the flexibility of the membranes by preventing the fatty acids from becoming too packed together. Questions on the fluid mosaic model? Mm-hmm.
Just like kind of like a quick question. Are the peripheral proteins only on the inside?
No, they could be extracellular peripheral or on the inside. It's just they're not in the membrane. They're just like loosely attached to one of the sides. But they could be on the outside, they could be on the inside.
Okay.
Okay? But they are on the periphery, they're not through the membrane.
The proteins that don't like go all the way through,
Mhm.
like the one in the middle, would that be?
This one,
No, over.
like this one?
Yeah, that one. Would that
Yep.
be integral or peripheral?
This is an integral protein. Okay, it is integrated into the membrane, but it's not a transmembrane protein. This one is a transmembrane. It spans the whole membrane. All right, so all transmembrane proteins are integral proteins, but not all integral proteins are transmembrane proteins. Okay? I think I said that correctly. So we went over the fluid mosaic model. Don't write this down. You already did. Okay? I'm going to talk about patches. Here's your hydrophilic head and your hydrophobic tails, right, which gives you a hydrophilic regions of the membrane and hydrophobic regions of the membrane. This is showing you the structure of the phospholipids, the tails. Why is this one bent? Why is this one right here bent? Brett.
It's got the double bond, so it's unsaturated.
So it's an unsaturated fatty acid. Why is that important? Think about the structure of a membrane.
This just creates a kink.
It does create a kink, and what does the kink do? It makes it like liquid. Why? It doesn't stack easily, right? The straighter, the saturated the fatty acid, the straighter the line of the fatty acid tail. The straighter the line, the less space it takes up, and you can pack them in all together, right? When you get a kink, it prevents it from packing in too close, and that's why oils are mainly unsaturated fats, okay, and they are liquid at room temperature. That helps to keep the fluidity of the membrane as well, okay? Oh, look at that. That's a nice segue into the fluidity of membranes, all right? Fluidity in terms of movement, fluid. It's changing. Okay? This is the lipids that make up the membrane can move. The proteins that make up the membrane can move. They can kind of float to different parts of the membrane and move around as necessary. Okay? As they need to be. Now it says up here that the lateral movement's lateral. Okay? So if here's our phospholipids. Right? Moving this way and that way is okay. It's quick for the lipids, right? Moving side to side, left to right. Why is it harder to go from here to there? Flipping.
Because of the polarity.
Because of the polarity. In order for this part to flip, right, to turn, you would have to put two polar regions, two polar pieces would have to go through a hydrophobic area in between. That's very difficult to do, right? Flipping back and forth. There are enzymes that catalyze that reaction and do it, but it is not as easy as moving lipids side to side. They're on the same plane. There's no change in polarity. They can float left to right rather than flip top to bottom. All right? Why are the proteins a little more slowly? Why do proteins move slower than lipids? They're bigger. They just got bigger, and they have parts that are encompassed within a hydrophobic region or wherever it might be, so they have to go slower. And most of the time, they are in a directed manner. Right? There's some direction. We're going to cluster proteins together for something, or we're going to, whatever it might be. So we're going to direct that motion. Right? If something's difficult to do, well, you want to be efficient about it and only do it if we have to. Okay? So here's an example. Membrane proteins. So in culture in the lab, you can take a mouse cell that has labeled membrane proteins that are fluorescent red, right? And take a human cell with membrane proteins that fluoresce in green. And you can actually stimulate these two to merge together. They are similar in structure in terms of a phospholipid bilayer. Okay? And when you catalyze that reaction, right, by adding it in a certain aqueous solution with certain chemicals, it happens. Okay? But what you end up seeing is that the human green fluorescent proteins and the mouse red fluorescent proteins, they don't just stay in one area, they float and they diffuse, right? They move so that they're evenly spaced, and you end up with this mixed proteins after about an hour. Okay? This is a great example of the fluidity of the membranes. If membranes weren't fluid, what would happen? If there were no movement allowed, what would happen to the proteins? Nothing. You'd have this big cell, it would look like this, right? And you'd have all the red proteins over here on one side, and you have all the blue proteins on the other. And they wouldn't move. If they were stuck in place, that's what would happen. And you know what? Some of the proteins are stuck in place. All right? But a lot of them, most of them, have some type of fluidity so they can move about wherever they need to. All right? The other thing about membrane fluidity, okay, we talked about cholesterol and helps to keep those phospholipid tails from becoming too tightly packed. All right? And when temperatures cool, those phospholipids get packed in tight. So when you get too cold, right, it gets too cold, that the membrane becomes more viscous. It's harder to move around, right? It's not as fluid. Think about sticking, we all love bacon, right? How many of you have cooked bacon at home? When it's warm, right, warm bacon fat, it's basically oil in the pan, right? But when it cools down, what happens to it? It gets become a solid, or it gets harder, right? A semi-solid. That's because they're saturated fats that are being pushed in together, right? Becoming closer, and the type of lipids will dictate what temperature that membrane becomes a solid. Okay? So, membranes need to be fluid in order to work the right way. Okay? If not, they don't do transport as well. They don't maintain the boundaries as well. It's difficult for the cell to do its job. All right? And they are usually about as fluid as salad oil, okay, or as vegetable oil. Okay, so this is what it looks like. Fluid. What do you notice here?
The tails are very bent.
The tails are bent, which gives what? Space. So if we were to count, excuse me, how many phospholipids are here in this area, what will that give us? A density, right? Density of molecules in an area. Which one's more dense? Which one's more dense, 1 or 2? 2. Okay? So, more dense means higher viscosity. It's thicker. There's lots of stuff to go through. So now if I wanted to transport, I don't know, let's see, something that diffuses across the membrane, let's say oxygen, across the membrane in number 1 versus the membrane in number 2, which one's going to be easier? 1, why? There's just less stuff to go through, Brett.
So what keeps water from flooding the inner layer when there's space?
When there's space here, the polarity of water keeps it out.
So even though there's space, water to fill it won't?
No. Right, the polarity of water will keep it out. All right? So now you have not only the unsaturated tails prevent packing, right? But also now, even if we have, let's compare 2 and 3. Which one's more dense? 2 or 3? 2. So cholesterol now has embedded in here and prevented packing so much, right? So there's a combination of unsaturated fatty acid tails and cholesterol that work together to help maintain and prevent this from happening. You prevent the situation in number 2 from happening, and you maintain the structure of the membrane, and you maintain its most efficient function in transport and boundary. Okay? So not only unsaturated fatty acids, but also the presence of cholesterol help maintain the fluidity of membranes. Okay? And so at warm temperatures, cholesterol restrains the movement of phospholipids so you can't move left to right. All right? And at cooler temperatures, it maintains the fluidity by preventing tight packing. All right? But at both. So in some cases, it actually restricts fluidity, the movement of phospholipids from one area to another. And in other cases, and this is the more important case, at cold temperatures, it maintains fluidity by preventing the packing of the fatty acid tails. Questions on that? What, yes, can,
So all the fatty acids are unsaturated, it's just like the extent of how much kink there is.
so it depends. Not all the fatty acids are gonna be unsaturated, okay? And so the composition of the membrane, what are the fatty acids unsaturated, unsaturated? How much cholesterol is there? Really depends upon the species you're talking about, okay? So if you're talking about bacteria, their membranes are still made of phospholipid bilayers. They live in different environments than animal cells, right? So they might have a different composition of of their membrane than we do because of their environment, all right? Some bacteria or some archaea will also be different. Or some archaea live in vents or hot springs.
So would like any bacteria or any species, like would they have totally saturated?
They might. So if you find a bacteria that lives in a hot temperature, they might have completely saturated fatty acids because of what they need the membrane to do. So the structure changes based upon, the function. And that actually leads right into the evolution topic. Evolution of this membrane, the structure of the membrane is related to where they live. Okay? And where their environment around them will dictate how the membrane functions. Okay, and we know structure and function is related. All right? And there's actually an ability for you and for us to alter, and for cells to alter the composition of the phospholipids that are present depending upon what's going on. Okay? So if we need a more viscous membrane, we're going to add phospholipids that have saturated fatty acids. If we need a more fluid membrane, we can produce phospholipids that have unsaturated fatty acids. Okay? So temperatures vary actually lead to differences in membrane composition. So especially organisms, cellular organisms that might live in marine or aquatic environments that have differences in temperature. All right? As we get towards winter, it gets colder. The bacteria in Lake Massasoit aren't going to die off. They're going to respond to the change in temperature, the icing over, right? They're going to alter their membranes in such a way that they can adjust to the change in temperature. Okay? Now, membrane proteins. So that's the phospholipids. Let's talk about the proteins. There's two basic categories. They could be either embedded within the membrane or attached to the side. Okay? A lot of times these proteins are grouped together with related functions. All right? You might have a receptor that's linked to another interior protein that's going to relay a message, or there's functional grouping that occurs. So we talked about integral versus peripheral proteins. All right, so we don't have to go over this. Integral proteins span the whole membrane, and that area that spans the membrane has to have hydrophobic regions of the protein in order to be stuck in there. All right, so this is what it looks like. You have most of the time, you see these right here. What are those? They are, Jesse.
Alpha helixes.
Alpha helices, right? And that's an element of secondary structure of the protein that usually span the membrane. That's the best way for proteins to span the membrane and kind of hide their hydrophilic regions in the middle and expose hydrophobic regions so that they're in that non-polar part. Okay, so you saw this is a 7 transmembrane protein, right? So 1, 2, 3, 4, 5, 6, 7. Right, it actually spans the membrane 7 times. Okay? But its structure is so that it can go back and forth. It's the same one. This is the same picture you saw at the beginning with the pretty colors, just in a different diagram. Sorry. All right. There are 6 major functions of membrane proteins. Number 1, transport. Absolutely number 1. The cell needs things that the phospholipid bilayer does not allow in. Right? The cell needs water. The membrane, the phospholipid bilayer by itself doesn't like to let water in, but the cell needs it. The cell needs glucose. Glucose is too big to fit through the phospholipid bilayer on its own. Okay? The cell needs amino acids. The cell needs lots of stuff that on their own do not pass through the phospholipid bilayer. So the proteins that are there, that are integrated into the membrane, they work to transport those necessary substances in and out of the cell. All right? With that, there is also some enzymatic activity. They can break things down, build things up, all right? Hydrolyze ATP for whatever reason, and go from there. There's attachment to the extracellular matrix. We already talked about that one. Integrins, right? We talked about cell junctions. Cell-cell recognition. Recognizing what's you versus what's not you. Okay? We'll talk about blood groups when we talk about that. All right? Intercellular joining, we talked about cell junctions last week, right? Right, so formation of tight junctions and gap junctions and desmosomes. Those are all linking of membrane proteins that come together to hold cells together. And then finally, signal transduction. Signal transduction is defined as transferring a cellular signal, a cellular response, from the outside of the cell to the inside, or from the membrane to the nucleus. Those are your major functions. Okay? So transport, move things in and out. Lots of times we hydrolyze ATP. Enzymatic activity, this membrane protein is going to break down this substance and produce another substance. Okay? Attachment to the extracellular matrix. We already chatted about that one last week, so we don't need to do that one. Cell-cell recognition, antibodies. Okay, antibodies. White blood cells walk around and they recognize the carbohydrates, the glycoproteins and the carbohydrates that are sticking up on cell membranes. And all of your cells have a ID card, and that's what these little carbohydrates are. They determine, like, that's you versus a cell that's inside of you that's not you. Okay? And if does not recognize this, recognizes it as not you, it will develop an immune response. Intercellular joining, and then you get signal transduction. Many integral proteins function as receptors. Okay? This is a receptor that will bind to its ligand, or the molecule that it's specific to. And then once that it's bound, then that starts what my graduate professor described as a cascade of intercellular events. Stuff that happens inside the cell from there. Okay? But the point is, the signal is going to be relayed from the outside, here's your receptor, from the outside to the inside, eventually to the nucleus. And that's going to change the way the cell behaves. Okay? Okay? So we're going to go through a couple of these. We're not going to go through extracellular matrix because we just did. We won't go through intercellular joining. We'll talk about a couple of them. Okay, one of the easier one, cell-cell recognition. And we're going to talk about carbohydrates are important in that process. You have glycolipids and glycoproteins. All right? So you have the cell membrane, right? And you have a protein on the cell membrane. That protein, okay, may or may not have a carbohydrate chain on the outside. Okay? So, in many cases, there are these side chains of carbohydrates that are linked to the R groups from amino acids within the proteins. So it's like a skin tag, like a little ID tag or something like that. Okay? And in some cases, you have one of the phospholipids, all right, that could have a carbohydrate that's linked to it. All right? So here's your glycolipid. This is your glycoprotein. Glyco refers to sugar or carbohydrate, and then what it's linked to, either a lipid or a protein. All right? And those carbohydrates are what determines what's you versus not you. Okay? How many of you know your blood type in here? All right, what's your blood type? O positive. Okay, what else?
O negative.
O negative. What's yours? AB positive. Awesome. Okay, because the O's, they don't really count. Okay? So you have, there's a possibility of 4 blood types. A, B, AB, or O. Okay? The A and B, all it tells you is what carbohydrates are on your red blood cells. All right? So you have, here's your red blood cell, right? Let's minimize this thing. Here's your red blood cell. Happy, happy. Okay? If you are A, you have one type of carbohydrate on there. If you're B, you have another type of carbohydrate on there. All right? In the case of AB, you have both. Here's A, here's B. That's it. So the carbohydrates dictate what your cells are. So we know that AB is different than O, right? Here's O. They don't have any. Okay? And A blood cells have A. All right? Now, the crux of all this is that if you don't have the carbohydrates, your body produces the antibodies. The opposite antibodies. Okay? So here's O. Call that A, and this is AB. All right? Now I said, if you don't have the carbohydrates, your body produces the antibodies. Right? So in the case of O, they produce antibodies against A and antibodies against B. Why would they produce antibodies against A and B? Because it's not them. It's not me, right? So if I run into a cell that has this A carbohydrate that's not supposed to be in my body, I'm kicking it out. Brett.
What about that super rare blood type? I think it's R.
No, there's RH factor.
That.
RH is another one. Same thing, it's just another one. Either you have it or you don't, and a lot of times you have to be exposed to it to have it. So, but is another carbohydrate moiety, that's a carbohydrate chain that's on the side of blood vessels, or blood cells. Okay? So now, O, what do you think, what antibodies does the person who has AB blood produce? In their own blood? None. None. Okay? Because if you produce an A antibody, this is going to react to that, and that's going to cause the cells to break down. Not good. If you produce the B antibody, same thing, breakdown. No good. Right? Okay. So this is why we call O-type blood, the universal donor. So I could take the red blood cells and give it to this guy. It will never be recognized as not you. He doesn't have a B-type carbohydrate. I can give it to this person and won't be recognized as not you because I don't have an ID card. That carbohydrate that your immune system is looking for is not there. So it's not going to have a response. This is the importance in cell recognition. So that one cell is going to recognize what's you versus what's not you, what type of cell this is versus what type of cell that is, where it's supposed to be. Okay? Every cell in your body has a little ID card. It has a barcode. Okay? That tells you what's what. Yes? Positive is the Rh factor. That really is so O positive, that means you are O-type blood, positive for Rh. O negative, you're O-type blood, negative for Rh. Rh is another carbohydrate that's there. Okay? And that, when you talk about blood in human anatomy next semester, you will talk about what that means in terms of, it's really important when you talk about newborns or fetuses that are developing, and a mom who's Rh negative and the fetus, or the mom who's Rh negative and the fetus which is Rh positive. That causes problems. Okay? Question, we'll skip this insidedness of membrane, and we'll stop there for today. Okay? The videos are up. Go through the videos, because from here, selective permeability, I will finish chapter 5 on Wednesday. Okay? We're going to talk about passive transport, diffusion, facilitated diffusion, osmosis, active transport, and endo and exocytosis on Wednesday.
So it's our exam 5, 6, 4, 5, 6, 7, 4, 5, 6.
There'll be part of 3, 4, 5.
Okay.